Hi everyone, I am Mariam Islami, medical doctor, PhD of genetics, and fellowship of regenerative medicine from Harvard Medical School. Today I would like to talk about molecular genetic techniques for you.
Last session we talked about DNA, what is gene, what is chromosome, and we understood about Human Genome Project. Today we want to know about gene technologies such as DNA sequencing, gene expression, gene mutation, gene variation, gene profiling, recombinant gene, gene engineering, and transporter gene and cloning.
Here you can see some images about the DNA and chromosome. This is the nucleus. You can see chromatin here. Chromatin and condensed chromosome structure here. You can see here the double-strand DNA, the histones that are some proteins. Here we have chromosome, the arm of chromosome, centromere of chromosome, telomere of chromosome. Here we have some bases such as cytosine, guanine, thymine, cytosine, and adenine. Here we have double-strand DNA, and this is the RNA, single strand. Here we have the bases, the sugar and the phosphate. We call this nucleotide. And again, DNA, nucleotide, chromatin, and chromosome. Again, you can see double-strand DNA here. The protein, we call these histones, chromatin, and again chromosome. You see these from DNA to chromosome and the sequences of these bases such as adenine, thymine, guanine, adenine, and cytosine. Here you see the duplication of DNA. With duplication, we will have another DNA, and this is replication, that we have RNA from DNA with transcription, and after that, with translation, we will have the protein from DNA, RNA, and then we have the protein, and tissues are from the protein. Here again, you can see DNA, RNA, and proteins, and these are chromosomes. This is XY, it means the male, and this is XX, that this is female. You can see a karyotype, that is all the chromosomes of a man or a woman, that you can see here.
So what is gene technologies? Gene technology is defined as the term which include a range of activities concerned with understanding of gene sequencing, gene expression, gene variation, modifying genes, and transferring genes to new hosts. Genes are found in all living organisms and are transferred from one generation to the next. Gene technology encompasses several techniques, including marker-assisted breeding, RNA, and genetic modification. Only some gene technologies produce genetically modified organisms. We use the most appropriate technique or combination of techniques to achieve the desired goal. DNA sequencing is the process of determining the nucleotide acid sequence, the order of nucleotide in DNA. It includes any methods or technology that is used to determine the order of the 4 bases: adenine, guanine, cytosine, and thymine, that you can see in this picture. The advent of rapid DNA sequencing methods has greatly accelerated biological and medical research and discovery. Knowledge of DNA sequences has become indispensable for basic biological research and in numerous applied fields, such as medical diagnosis, biotechnology, biology, virology, and biological systematics. Comparing healthy and mutated DNA sequences can diagnose different diseases, including various cancers, characterize antibody repertoire, and can be used to guide patient treatment. Having a quick way to sequence DNA allows for faster and more individualized medical care to be administered and for more organisms to be identified and cataloged. The rapid speed of sequencing attained with modern DNA sequencing technology has been instrumental in the sequencing of complete DNA sequences or genomes of numerous types and species of life, including the human genome and other complete DNA sequences of many animal, plant, and microbial species. An example of the result of automated chain termination DNA sequencing. The first DNA sequences were obtained in the early 1970s by academic researchers using laborious methods based on two-dimensional chromatography. Following the development of fluorescence-based sequencing methods with a DNA sequencer, DNA sequencing has become easier and orders of magnitude faster. So, what is the application of DNA sequencing? What is gene mutations? Gene mutation is any change in the genetic information of any organisms. These changes occur at many different levels and they can have widely differing consequences. Mutations may be caused due to effects of physical agents and chemical agents. Mainly, there are different types of mutations, which include deletions, insertion, point mutations, substitutions, missense mutations, nonsense mutations, and etc. Most disease-causing gene mutations are uncommon in the general population. However, other genetic changes occur more frequently. You can see here some kind of mutations. You can see substitutions. You can see insertion. TAT, insert here. And you can see here deletion mutation, that C and A are omitted. So we have another term, gene variation. What is gene variation? Genetic variation refers to diversity in gene frequencies. Genetic variation can be referred as differences between individuals or to differences between populations. Mutation is the ultimate source of genetic variation, but mechanisms such as sexual reproduction and genetic drift contribute to it as well. Genetic variation is based on the variation in alleles of genes in a gene pool. It occurs both within and among populations, supported by individual carriers of the variant genes. Genetic variation is brought about by random mutation, which is a permanent change in the chemical structure of a gene. You can see this here, some kind of mutation. You can see deletion on the chromosome, duplication, insertion. See here, we have insertion for chromosome 20 that comes from chromosome 4. And also here we can see translocation between chromosome 4 and 20. These are some examples. And now, next generation sequencing, or NGS, is also known as high throughput sequencing. It is a term used to describe a number of different modern sequencing technologies, including Illumina sequencing, whole exome sequencing, targeted sequencing, amplicon sequencing, exome sequencing, de novo sequencing, transcriptomics, and etc. These recent technologies allow us to sequence DNA and RNA much more quickly and cheaply than the previously used Sanger sequencing, and as such have revolutionized the study of genomic and molecular biology. Now we should know what is gene expression. Gene expression is the process by which information from a gene is used in the synthesis of a functional gene product. These products are usually proteins, which functions as enzymes, hormones, and receptors. Look at here, we have a gene, you can see RNA, and it translates to protein that function as enzyme, hormones, receptor, or some tissues. So genes which do not code for proteins, such as rRNA or transfer RNA, code for functional RNA products. Gene expression is the process by which the genetic code, the nucleotide sequence of a gene, is used to direct protein synthesis and produce the structure of the cell. Genes that code for amino acid sequences are called as structural genes, that you can see one of them here: DNA, RNA, and protein. Now we should know about gene profiling. Gene profiling is the measurement of the activity of thousands of genes at a time, or to create a complete picture of cellular function. Gene expression profiling is determination of the pattern of genes expressed at the level of transcription under a specific circumstance or in a specific cell to give a global picture of cellular function. In almost all cells in an organism contain the entire genome of the organisms. Only a small subset of those genes is expressed as mRNA at any given time, and their relative expression can be evaluated. Techniques which are involved in profiling include DNA microarray technology and sequence-based techniques such as a serial analysis of gene expression. You can see expression profiling with DNA microarrays in this picture. We want to know about another technique: real-time polymerase chain reaction. A real-time polymerase chain reaction is a laboratory technique of molecular biology based on the PCR, polymerase chain reaction. It monitors the amplification of a targeted DNA molecule during the PCR in real time, not at its end, as in conventional PCR. Real-time PCR can be used quantitatively and semi-quantitatively. Two common methods for the detection of PCR products in real-time PCR are: 1.